Dear students, Namaste. Let us discuss the new experiment under the course or under the subject Pharmacology Practical. Experiment number 3, commonly used instruments in experimental pharmacology. In previous experiment, that is experiment number 2, we already discussed what are the different laboratory animals which is used for experimental pharmacology for the purpose of the drug evaluation or to study or bioassay or screening of the different animal models with help of different drugs. Now in this experiment, what are the different common instruments which is used for practical pharmacology or experimental pharmacology? Let us discuss one by one in this video. Till you not subscribing my channel, please subscribe and press the bell icon for the future updates. And myself, Mr. Abhijit Ankush Katre, working as a lecturer in Keshiris Krishna College of Pharmacy, Karnataka. So let us discuss what is the aim of the today's practical commonly used instruments in experimental pharmacology. What are the practical outcomes for today's topic or today's discussion? What are the practical outcomes? Practical outcome number 1, the list of various instruments and equipments used in experimental pharmacology laboratory. What are the instruments? कौन से instruments हम use करते हैं? Equipments कौन से use करते हैं? Let us find out or let us discuss in this experiment. Second outcome is describe the Sherrington rotating drum machine, organ bath assembly and various levers which is used for the practical purpose. So after completion of this experiment, learner or the student will be achieve these two outcomes. Let us discuss what are the instruments or the apparatus which is used for the experimental pharmacology. So number 1, organ bath assembly is also known as student organ bath assembly. Second is Sherrington rotating drum and Camograph. It is also called as Camograph Paper, which is sticked into the drum. Number 3 is recording levers. Number 4 is different instruments used for study animal models. So these are the 4 different instruments, equipments or the apparatus which is used for pharmacology. You pause this video and write in your practical notebook. These all are the very important for your viva as well as your synopsis and practical purposes. Now let us discuss one by one, and this is the first is the organ bath assembly. What is the use of this organ bath assembly? Used to study the effects of drugs on tissue. And this is the instruments is known as organ bath assembly or student organ bath. It contains different parts or different 5 parts. Number 1 is the organ bath. This is outermost rectangular square box, plastic square box is known as organ bath. Into that there is a presence of organ tube where the organ is isolated organ is inserted or into this kept into this organ tube. Then inlet outlet, with the help of inlet outlet we can add or remove the fluid or the water into that organ bath or organ tube. Aeration tube, which is provide the air to the isolated muscles to allow the that specific isolated tissue or the muscle throughout the experiment. We can supply the air with the help of aeration tube and reservoir. Glass foil is also present to maintain the water temperature as neutral human body temperature. So this is the first instrument known as organ bath assembly. Second one is Sherrington rotating drum. That drum machine is used to check the movement of camograph at desired speed. If you see this image, it's containing circular, rectangular, cylindrical in shape, not rectangular, black spot. On this black image, we stick the camograph paper. paper, and with the help of that Camograph paper we record the readings. And this drum is divided into three parts: dry, recording drum, and the speed gear. These are the instrument known as sedentary rotating drum. This is drum and this is device. This is dry, this is recording drum, and here is the gearbox or speed gear. Then another equipment or apparatus used to experimental pharmacology is Camograph paper to record the recording from the student or members. This is graph paper. That paper sticked on this drum, and with the help of desired speed we can record the responses of the tissue or the isolated organs or the isolated muscle part. The part number 3, before going into the part number 3, this is the actual image where the Sherrington rotating drum and organ bath assembly is fixed or arranged. And this is organ bath. Here is the organ bath assembly, and here is the organ student, sorry, not student, Sherrington rotating drum. And with the help of the lever, we can record the reading with the graphical response or response curve. So this is the image. You take the screenshot or snapshot of this image and draw in your practical notebook as well as your manual if possible. You can get the idea about what is the arrangement before starting the experiment. Now let us move to the next part. Part number 3 is levers. This is the actual image which is taken into from this lab requirement arrangement like this. This is student organ bath. This is Camograph which is sticked on this center rotating drum, and this is stainless steel small wire is known as lever. Then what are the levers? There are different 4 type of levers which is enlisted in your experimental syllabus of pharmacology. The recording levers, number 1 is a frontal writing lever, second is sample lever, number 3 is the Sterling heart, and fourth one is gimbal lever. This is all very important for your spotting as well as viva purpose. Enlist different type of recording levers. These are the levers. Frontal writing levers generally used isolated tissue preparations. Sterling heart levers generally used isolated heart organ preparation experiments, and the gimbal and simple levers are generally used for specific experiments. So these are the recording levers. And how you check the magnification response with the help of magnification value or the formula. This is the magnification graph. This is the structure of lever. How to calculate this magnification value? With the help of distance between the fulcrum and stylus divided by distance between fulcrum and the writing point or point of attachment to the tissue. This is fulcrum where the lever is fixed. From the left side and from the right side, you balance this distance with the help of this fulcrum. Means the x and y is the ratio. We can balance that lever with the help of this ratio. Now next part is what are the different instruments or apparatus which is used in pharmacology practice. Total 8 apparatus used for these experiments. Elevated pulse mass apparatus. With the help of this we can check the anti-anxiety effect. Open field also same. Actophotometer, with the help of actophotometer we can check the hypnotic and sedative effects. Telflick apparatus and hot plate method used to check the analgesic activity. Protarad is also used to check the muscle relaxant activity. Electroconvulsometers used to check the convulsions or anticonvulsant effects. Pole climbing apparatus which is used to check the timing effects in the animals. So these are the different 8 instruments, and here is this image. of all these 8. Elevated pulse mass, open field test app, Telflick apparatus, actophotometer. You pause this video and watch these all images, which is very useful for your spotting. The next one is the hot plate method, rotarod, Cook's pole climbing apparatus, and electroconvulsometers. So these are the different instruments. I hope you understand this all. If any doubt, you message me in description box. You can ask me any of your doubts with the help of this PPT. Watch this, and if any doubt, message me in description box. So thank you so much for the patience listening. Thank you once again.